We are in the mammal collection of the Museum of Vertebrate Zoology. Behind us in all these cabinets are the bulk of the mammal collection. It starts in one corner and goes up and down the aisles and all the way to the back. A little bit different from the bird collection. Our specimens are arranged taxonomically, and then our skeletons are put at the end of each family, whereas the bird collection, they are put all in one place. The bulk of what is in the cabinets here are our smaller skins and skull collections. This is our standard skin and skull preparation. So that rat that I just prepared downstairs will end up in a drawer like this with other Rattus rattus, accompanied by a clean skull eventually. So it'll take some time for us to do the curatorial work to catalog the specimen, send it back down to the prep lab to be cleaned, have it come back up, numbered, labeled, and then installed in the collection. But hopefully in about 4 or 5 months, that rat that I prepared will be in the collection and online on our database. So this is what the skin looks like. As you saw in the lab, it has a cotton body, wires in the feet and the tail. And then most importantly, it has a tag on it that has all the original data. And all of this data is available on the online database, ArcDivs. And then the skull for it is adjacent to it, and these are labeled. So this is a large part of our collection, the skin and skull collection. A smaller portion of the collection is skeletons. And so this is a complete skeleton of a muskrat. And this one has all of the bones to it. But in some cases, we save the skin and the skull. The bones of the feet stay in the skin, and then what we have left here is just a partial skeleton. That's another kind of representation. Then we also have fluid preserved animals. So with the herpetology collection, the vast majority of their collection is wet like this, but for us it's probably about 5 to 10% of the collection. And these animals are preserved as fresh as possible in formaldehyde and then transferred to 70% ethanol. And then in some cases, as you can see here, there are carcasses like that with the fur on them. Then there's also carcasses. So sometimes we'll remove the skin to make a study skin, but we'll preserve this. So if anybody wants to study muscle structure, internal anatomy, reproductive anatomy, stomach contents, anything like that, it's available to them. Occasionally we do find parasites in the animals. And so this vial holds stomach worms from another rat that was prepared. This one was from San Rafael in Marin County. And so this is going to go off to another home. And here are tapeworms from another kind of wood rat. And then also we occasionally find ectoparasites. So these are lots of ticks and fleas off of a mammal. This is probably something large like a deer. The ectoparasites will be cataloged here as a part, and then these will be given to the Entomology Museum. And then they eventually will give us back their catalog number, and we can put that in our database. So if anyone wants to check on the host for more parasites, or check the locality data or something like that, that's available. We have some other preparations in the collection. We have a small number of cleared and stained specimens. These are some bats that were cleared and stained. And this has been done for a variety of reasons. One is developmental biology, to see how different species of animals develop in their skeletons and their cartilage. Another thing that was done much more in the past was saving of cheek pouch contents from animals that have cheek pouches, like kangaroo rats. So this is a part of our collection that isn't online yet, but I've been working with students, some volunteers, and some URAP students to inventory the material, and then we'll eventually add it to our database, and then a researcher would be able to find them and analyze them. So hopefully you'll be able to see what these look like. These are grass seeds or other kinds of seeds found in the cheek pouches of kangaroo rats that these two individuals would have been bringing back to their burrows to stash for another time. But from these, you could eventually figure out the diets of animals.